Hello, welcome back to the Computational Genomics Lab. We're going to start working on our ATACseq NF core pipeline results, and those are in peak files. And so we're on level 8, and we're going to do lesson 803. So in lessons 08 ATACseq pipeline, 03 Iranges. Okay, so in this class, we are not going to actually load in our peak files yet. I know you're excited because you just walked through them in the last video, but before we do that, we have to think about a new type of logic. In this case, all of our peaks have a start and a stop, and that's a typical computational thing that's used in R called Iranges. So you can have a list of start and stops in a position coordinate. So if it was on a track, like a track and field track, you could say there's a position that starts at 0 and goes to 100 meters. You could have another position that's like starts at start and goes to 200 meters, 12 meters, whatever you want. So you can just have a start and stop. And it could be 12 meters to 100 meters, start, stop. And so the problem with the genome is we have chromosomes, and starts and stops could overlap on different chromosomes. But we'll deal with that later. First, let's learn some of that start-stop logic. Okay. So the package we're going to need for this today is called Iranges. It should be in sort of base R. And then in the next class, when we add the genome to it, we'll use Granges or Genomic Ranges. Okay, so the goal today is just to use Iranges, and there's some more detail on what I was just sort of describing of why you would want starts and stops. This part here on line 33 will explain why we need a thing called Granges, which is just a modification of Iranges. And so we're going to want to just load this for the sake of it for today. And if you want the manual, there's a manual here. We don't need genomic ranges today. Okay, so here is Iranges. There's a bunch of information on how it works, and we're going to go through some examples here. Okay, so let's make our first Irange. And so the only function we need to know for this guy is this function called Iranges with capital I, capital R, ranges, and what that function needs, because that's what's inside the parentheses. And we're going to name this first object IR1 or Iranges1, and use the Iranges function, and we're going to say the start equals, and we're going to say we're going to have 3 intervals or 3 people on the track. Okay, so the first interval is going to start at the 1 meter line. The second interval is going to start at the 200 meter line, and let's just say the track was more long than it is, 1,000 meter. So those are just 3 different starting positions for things. We don't know the ends will come in the next one to match them for a start and an end. So you might be thinking, well, why not just do start and end right next to each other? And you could do that. This is just a sort of faster way, especially if you start to have lots and lots of ranges. But really, we're just trying to understand the fundamentals of what is starts and stops, and then how do we do overlaps with them. That's the more useful thing. Okay, so if we do that, then we're going to say the stop for the start on 1 is 30. So we have an interval from 1 to 30. We have an interval from 200 to 299. And we have one from 1,000 to 1,200. So none of these intervals overlap. 1 through 30 is less than 200 to 299, and 299 is less than 1,000 to 1,200. So you can see it's just a little bit, you'll get really used to this, but I can see why it's a little confusing at first. Okay, so let's clean my environment here, and let's look at make our first Irange. Okay, so now I got an environmental variable with those 3 positions, and it's a formal class Irange, and let's take a look at it here. Give it a little more space. Cool. So our first Irange is here, and this will probably make more sense to you. So first Irange starts at 1, goes to 30, and it's 30 long. So Iranges also calculates the width, which is basically the stop minus the start. Okay. Second one goes from 200 to 299, and it's 100 long. Second one goes from 1,000 to 1,200, so it's 201 long. Okay, so that makes it look a little more thing. You can index into this and just grab the start, end, or width. So you can say, I want the starts for this, and I'll tell you those start values, the end values, and you can just get the width if you want it as well. with these start, end, and width functions. And those are not Irange functions. Those are just kind of base R functions. Okay, so let's make our second Irange, which is going to start at 17, end at 49. So that will overlap with the IR1. We're going to call this one IR2 using the Irange function. And the start is equal to these three values, with the C meaning paste these three values. And so 17 to 49 does overlap with 1 through 30, which we have in IR1. 100 to 199, which does not overlap with our 201. And 1100 to 1201, which will have some overlap. Okay, so that's kind of where we're getting at. We have two sort of Iranges now. We can ask where those positions overlap. And if you just think of those positions as peaks, you could start to make sense of, like, in sample 1 and sample 2, do these two peaks overlap? And you can get an answer for that. In just a second, we'll do that. Okay, so we can take a look at IR2. Oops, I didn't run it. We can take a look at what that makes, and we can see these three ranges. What's also cool is you can merge them. So we did, this is IR2 and IR1 looked very similar. They just have different starts and stops and widths. A similar format though, and so if we use C again, it'll paste them together and concatenate them into one big Irange. So let's say you had a peak file and another peak file, and you said, oh, I want to make them the same thing. You can just concatenate them, or into the same file. Okay, so let's make IR3 the merge of those two Iranges. Now we can see we have 6 entries with the different starts, stops, and widths. Okay, so what we can do is make names instead of having these 1 through 6. We've done these call names, row names to calls, and we can name the IR1 by pasting in replicate 1. So let's just say that we had 2 replicates of ATACseq, and we wanted to understand which peaks overlap between the two of them. So we can use, this is paste for common text with quotes on it, replicate 1, peak 1 through 3, and the separator is a space. So that's going to give me the names for IR1, which currently, if we run IR1, does not have names there. So let's do that. And that information here gets piped right into this names function, which is base R. Okay, so let's take a look at what happened to our IR1. Awesome. So we see this replicate 1, peak 1. And so we can make a peak file, and that's kind of what we're doing, is we're learning on just with a few peaks and a couple replicates how we would do this with the whole genome, which might have 30 to 50 to 100,000 peaks. So starting simple here. Okay, so let's make the same thing for IR2. There we go, and let's take a look at it now. It has replicate 2. And now we can merge the two and take a look, and we can see now they're named, which helps us a lot, because when we did that merge previously, they didn't have any names. Okay, so now you can see how you can name your things and merge ranges together. So really it's just a file, a list, if you will, of starts, stops, and widths. And so now we're going to look at the overlaps. So we have this IR1. Or I think, yeah, we're just remaking it here. If you want, let's just do that for the sake of starting over. So now IR1, looks like it used to with no names. And IR2 will now look the same as we started with. So there's no names on there. Okay. So OV1 stands for overlap 1, and let's find the union between IR1 and IR2. So we have these two ranges. So each sample had 3 peaks. They had different starts and stops, and then we're going to ask if how many of the 3 peaks overlapped between the 2 samples. To do that, we can use union and or intersect, and we're going to look at both of those and why there could be some things to think about there. Okay, so with union, we're going to say all the overlaps. We're going to take a broad thing. With intersect, if it's like this, it's going to say it only overlapped here. Okay, so let's do union is the function we're running. Base R again, and IR1 and 2 are going to get merged. If you want to look ahead, you can see which peaks are most likely. It's a good thought exercise to see which ones are going to overlap and which ones are not. Okay, so that's what I'm doing here, is you can look at IR1 starts and stops, IR2, and try and do some matching if that is helpful to you. But let's look at OV1. Okay, so OV1 now says 1 through 49 was the first one, 100 to 299. So all three had an overlap. So the problem with that is that if we look at IR3, which has all of our data, what it said was between these two samples at position 1, we had an overlap from 1 to 30 with 17 to 49 is where the overlaps occurred. And so what it does is it takes, with the union, it takes the furthest start and the furthest stop, so it makes like one big long peak. And so that's where the 49 came from. When we look back up here, that was the first one. The second overlap is this 200 to 299. They don't overlap. So this is why I'm pointing this out, is that union can be kind of dangerous, because what you're doing is you're saying, okay, give me the range in sample 1 that has a start, and I'll give you the furthest stop in the next range. So you probably don't want to use union, and so that's just why I wanted to show you that. So let's try intersect, which is more of what you were thinking about, I'm guessing, if two peaks actually overlap. Okay, so now let's look, OV3. Here, we only see 2 entries because only 2 of those intervals should have overlapped. We had 1 through 30 and 17 through 49. And so in intersect, it takes the furthest start towards the end and then the closest end to the start. And that's 17 and 30. So the width of that peak is 14. And so it's defining the only parts that overlap. Whereas the previous one said, oh, there was an overlap here, but this peak started there and that peak started there. And it's useful for other things, but not in this case. So now we can see that 2 of those peaks overlapped, and you can go back and like try that, just write on a piece of paper like with the line and say, okay, this is 1 through 1,000, and here's a little slide, and here's one. And we'll take a look at them more in a second. Okay, or in the next video. Okay, so we can also count the overlaps. So here what we got is a new Irange from the overlaps of the 2 Iranges. And so we could use this other function called count overlaps in Iranges, which is going to count the overlaps. And what it says is when I test Irange 1 and 1, they didn't overlap. They do overlap. The second one did not overlap, and the third one did overlap. So that's the 1, 0, 1, meaning true, false, true. And so you might not need to know where the starts and stops were. You just wanted to know which were the 2 peaks that overlapped, and we can take a look at that with this IR3 here. And we can see that 1 through 30 overlapped with 17 through 49. This position did not overlap because 100 to 199 does not overlap any of these numbers. And the third one does overlap because 1000 to 1200 overlaps 1100 to 1201. And that's kind of the logic. If this is a nice example just to get the fundamentals down, and we're going to take it bigger and bigger and bigger as we go. All right, well, see you next time.